Good morning, everybody. Today we are discussing the topic bacterial growth curve. Okay, so I think everyone knows that bacteria divides by binary fission, and bacterial division is very important because when bacterial division occurs only, we will get bacterial colonization. So, in this, we need to understand what are different stages of bacterial multiplication. The bacterial multiplication occurs in 4 stages, that is log phase, lag phase, log phase, stationary phase, and decline phase. So, the bacterial multiplication occurs in 4 stages: lag phase, log phase, stationary phase, and phase of decline. The first stage, which is known as lag phase, it is the starting period, that is, it is the time period between the inoculation and the beginning of multiplication. The time period between the inoculation and the beginning of multiplication. Here the bacteria is not multiplying, but what happens is that the cell increases up to maximum size and it adapts into the new environment. So that is occurring in lag phase. Here the bacterial multiplication does not occur, but there will be adaptation of the bacteria and synthesis of new enzymes in the time period, and bacteria prepares itself for multiplication. The second stage, which is known as log phase. Log phase means the multiplication of the bacteria occur, and the cell will be multiplying in a geometrical progression, and this is a stage where the bacterial size will be smaller, and you can see the biochemical activities will be in this stage. This is a stage where the biochemical properties of the bacteria will be best, and if you do gram staining, you will get the exact gram staining picture of bacteria. So log phase is otherwise known as the multiplication phase. The population of the bacteria multiplies. The cells are smaller but biochemically active, and you get uniformly stained cells. The third stage is known as stationary phase. In the case of stationary phase, the number of new cells formed will be equal to the number of dead cells, so that the number of viable cells is equal to the number of dead cells that has occurred in the medium. And the speciality of this stage is that sporulation will be occurring in this stage. Bacterial toxins and antibiotics are synthesized in this stage. So in the case of stationary phase, what appears is that if you have 10 new cells, there will be 10 dead cells. So there will be an equilibrium between the dead cells and the new cells. And sporulation occurs during this phase. And not only sporulation, exotoxins also will be produced during this stage. And next step is, that is the last stage, is death phase. In the case of this stage, the bacterial multiplication does not occur. Whereas what happens? There will be exhaustion of nutrients, means the moisture and the vitamins and the nutrients present in the medium has been utilized by the bacteria for its multiplication and metabolic processes. And also there will be accumulation of toxic products and autolytic enzymes. So here the number of dead cells will be in more, and the number of living cells will be less. In the declines or the death phase of the bacteria, the number of living cells will be very, very less, whereas the number of dead cells will be very much higher. So these 4 stages are the stages of bacterium that is calculated by plotting a curve. Here a graph curve is plotted by taking into consideration the time and the number of cells formed during the time period. So such a curve is known as bacterial growth curve. So how did we get the bacterial growth curve? We are taking time plus the number of cells formed in the particular time, and that is plotted against the curve. So that is divided into 4 stages, that is lag phase, log phase, stationary phase and decline phase. And I told that in the lag phase there is no multiplication of bacteria, whereas in the log phase bacteria multiplies, and in the stationary phase there will be an equilibrium between the number of new cells and number of dead cells. And at that stage there is complete exhaustion of nutrients, and the dead cells increases and the number of living cells is very less. So these are the 4 stages of the bacterial growth curve, and there are certain factors which affect the growth also. That includes oxygen, carbon dioxide, nitrogen, pH, osmotic pressure, etc. But that I think where we all know that bacteria needs basic, such as requirement of oxygen is essential, carbon dioxide is essential for certain bacteria, where the pH of the medium is very important for multiplication of the bacteria. That is, usually the bacterial pH will be in 7.2 to 7.6. There are acidophilic bacteria and also alkali bacteria. And the temperature and light are also influencing bacterial growth. So in short, I can say bacterial growth will be influenced by a variety of factors that includes carbon dioxide, oxygen, light, pH, etc. And on considering on this, we are able to do the bacterial growth curve. So that is the topic we have discussed today, bacterial growth curve. I think all of you have understood. I request you to write an assignment on the bacterial growth curve, drawing the figure and submit to me in the next class.